Hi, my name is Sarah Wolf, and I'm a PhD student at Oregon State University studying marine microbiology.
Phytoplankton are marine plants who get their energy from the sun and require nutrients to grow. Bacteria get their energy by consuming waste material in the water column and use up oxygen in the process. In a coastal upwelling system like the Oregon coast, water that is low in oxygen but rich in nutrients gets brought up to the surface. These nutrients trigger a bloom of phytoplankton. The bacteria now have a plant-based buffet to indulge in. They eat up the waste in a frenzy and use up the oxygen. But when the oxygen levels fall, it can be harmful to organisms such as crabs, fish, or shellfish. This causes problems for the ecosystem and coastal communities who rely on the seafood industry as a source of income. When and where these coastal low-oxygen dead zones will form is difficult to predict. My research tries to identify the conditions that cause bacteria to create runaway low-oxygen zones sometimes and hit the brakes in others. I do this by recreating phytoplankton blooms and low-oxygen zones in the lab. Here, I'm going to show you a small-scale version of my experiments that we developed as a teaching tool for educational outreach. First, I collected and filtered water in Yaquina Bay on the Oregon coast to remove any larger organisms that would want to eat up our phytoplankton. Back in the lab, I added 3 different levels of nutrients to the seawater I collected and put those bottles, along with a no added nutrients control, in my east-facing office window to get some light. I tracked the results for 2 weeks, and our results show that the more nutrients added, the more dense the phytoplankton bloom. By creating controlled blooms in low oxygen zones in lab, I can look at how fast bacteria use oxygen, identify which bacteria are most active, and chemical signatures to help develop predictors to take back to sea. Interdisciplinary science, along with effective management, are necessary to better understand the cause, consequence, and potential mitigation of these low oxygen regions.